Okay.
So these are the supplies that you will need to perform this part of the test. Make sure to have your safety supplies, and anything that you use should be autoclaved or sterilized prior to use.
Yep.
Alright, so depending on the concentration of your sample, you may need to dilute it. With this one I have an undiluted sample, and with this one I'm going to dilute by 50%. If you're using a very thick sample like a return activated sludge, you may need to dilute it more, say 1 part of sample and 4 parts of dilution water. We use this 1X PBS phosphate buffered saline solution that has been sterilized to dilute the water, or to dilute the sample. With these samples, we need to disrupt the flux, disrupt the agglomerations that are in the samples. We use sterile syringe and sterile needle. These needles are 26 gauge. Be careful to not stab yourself with this. These needles are very sharp. So you can take your diluted sample and also your concentrated sample. You'll do the same process to both of them. And you just insert the needle and plunge the plunger of the syringe back and forth. This process is done for 10 to 15 minutes depending on the sample. Occasionally with a dirtier sample or a sample that has more particulates in it, you may get stuck and can't pull it up anymore, in which case just try to reverse directions. You may also get stuck while going down, in which case again just try to reverse directions.
Okay.
Alright, after you've done this for 10 to 15 minutes and your sample is well dispersed, it should look relatively uniform. Now at this point, depending on your concentration, we can put some of this sample onto our slide for later use. So I'm going to take this sample, which is diluted 2 to 1, and put a small amount on here. You can make a couple of different dots if you want. It doesn't really matter. Just a small amount is fine.
Go.
And after you've done your other dilution, you can do the same thing. In this case, this is an undiluted sample, and I'm just going to plate that here. The goal is to try and spread these out a little bit, but be careful to not scratch the slide. And then just let them dry. The clumps in there. After we've done that, we place it in the centrifuge. (6,000 RPM) for 5 minutes.
Rolling.
After the centrifuge has been run for 5 minutes, we open it and discard the liquid on the top. You can just pour it into the sink. I'll do that with both of my samples here. And now we fix these in a solution of 4% paraformaldehyde. This is frozen initially, and we warm it up by holding it in our hands while the centrifuge is running. And again, our 1X PBS. So we take 1 part of this and 3 parts of ice cold paraformaldehyde. This is our ice cold 4% paraformaldehyde. I have my pipette set to 0.4 milliliters. I'm going to use this 3 times, and then I'll do the 1x PBS one time for each sample. After you're done, this should go back in the freezer. Get a new sterile pipette tip. And again, 400 microliters of the 1x PBS. After you've added your paraformaldehyde and your PBS, you need to mix these well to make sure to get all of the cells in suspension. We don't want a clump like that. So you can use a vortex mixer, or in this case what I'm going to show is just fast mechanical agitation. Your samples might need more than this. Our samples, because they're coming from a synthetic wastewater, disperse relatively quickly, but it ought to look something like this with no big clumps when you're done. And now we take these and put them in the 4 degrees in the refrigerator and hold them there in the dark for at least 1 hour. Okay, it's been an hour now, so we're going to take these samples out. Nice and cold, and now we centrifuge them again. Again, 5 minutes, 6,000 RPM. Okay, it's been 5 minutes. We turn the centrifuge off, open it up, and again we pour off our clear liquid. Now we add 1 milliliter, 1,000 microliters with a new tip of our 1x PBS. This is to wash our cells. Again, we have the clump at the bottom, so we're going to vortex mix or manually agitate. After you've washed a second time, again, we dump off the excess liquid. For a final time. And now we store it in PBS ethanol solution. This is 50% PBS, 50% ethanol. And I have a new tip on. Always a new tip, always a new sterile tip. And one final manual agitation. Now these can be stored in the freezer at -20 for up to a couple of months. Okay, these samples that we put on previously are not yet dry. We need to wait until all of that water evaporates before we finish the sample preservation. And you can clearly see that there are a lot more cells in our undiluted sample than in our diluted sample right next to it. This means that our undiluted sample is going to be probably pretty difficult to quantify when we look at it under the microscope. Alright, so now our slide is dried. You can kind of see on our diluted one that it looks a little darker, or a little like there's more stuff on there. That's because our phosphate buffer has dried on there, and it's really just salt that has dried, so that's okay.
Yep.
Alright, so to ship this, what we would do is take the slide and just put it in one of these. So you just take a little bit of paper towel, kind of crumple it up, and then we put it in the top here to make sure that our slide doesn't roll around too much. You can also put multiple slides in here, put them back to back, for example,